Hi.
I just saw your message. Sorry, I was at the same time.
Yeah. Hello, Dr. Campisi.
Oh, no, call me Laura, please.
Oh, okay.
Hi.
Laura.
Hi.
Thank you so much for coming.
No, thank you for your interest in my work.
We'll start in around 10 minutes, so you still have some time to relax. We just opened the room earlier to make sure everything works and stuff. So.
Okay.
Katerina, please check the link to see if it works. I think this one works, hopefully.
Yeah, I think I have to compress it because it says you cannot preview it. So I think I made earlier a compressed version. Let me just add it to the Google Drive.
Okay. It's nice to have you with us, Laura.
Thank you. I try to understand how I can unmute myself in the meantime. Oh, I'm not really. I'm discovering this thing with you guys.
Oh, that's fine. The mute and unmute button is in the bottom right corner. You can see a microphone. When you click it, yeah, now
Got
you're
it.
muted. Now you unmuted. Yeah, so now you're muted, and when you click it again, you're unmuted. Hi, Jamie.
Greetings. Hello. How are you today?
Good.
Or this afternoon or this evening or whatever it is. Time zones.
We'll be starting in just a couple of minutes. I have everything set up now.
Very warm welcome to you, Doctor. So glad you could join us.
Oh, thank you.
Have you had a good day today so far?
Yeah, like in science, up and down.
that's a yeah, I like that. I like that. Okay, we're just sharing the room and everything, and we'll probably get started in a moment.
Okay, can you hear me or do I have too much background noise? Should Cecilia him do the introduction?
You're sounding okay to me. We're hearing some background sounds, but nothing overwhelming. Does anyone else hear
Yeah, no,
anything?
that's fine.
Yeah. Yeah.
Okay, perfect. So, welcome everyone to the Science Society, and a special welcome, of course, to Laura. Thank you so much for coming, and we really appreciate it. And for making the Clubhouse account and coming here, we really appreciate it. And
Thank you guys for your interest.
yeah, let me, before we start, let me introduce you a little bit to the audience. Dr. Laura Campisi, she's assistant professor in microbiology at the Icahn School of Medicine at Mount Sinai. And she did her Bachelor of Science at the University of Nice, Sofia, Antipolis, and her master's degree also at the University of Nice, Sofia, Antipolis, and her PhD in immunology also at the University of Nice, Sofia, Antipolis. And then later she did various postdoc positions, and the latest was at Mount Sinai Medical Center, where she is now an assistant professor at the School of Medicine. And yeah, she does really interesting research, and we are so honored to having you here sharing your amazing work. And before we start, though, with your presentation, um, Cecilia will ask you a couple of interview questions, if that's okay. And yep, we'll go from
Sure,
there.
sure.
Thank you so much, Laura.
Yeah, thank you so much for coming, Laura. Yeah, we'd just like to ask a couple of questions just so we can get to know you a little bit more. So the first question I have for you is just wondering, when did you discover your love for science, and how did you nurture that interest into what it is today?
I don't really know when my love for science started. I guess like my father is a doctor, a medical doctor. But I did, like in high school, I did the humanity. But I don't know. I always been curious, and I did 1 year of medical school. And when I was in medical school, I started thinking that I wouldn't like to be like a doctor, like in a hospital, but I would like to do research because I like better like everything that was like cellular biology, everything that has a mechanism in there. And so then I moved to the faculty in France because I'm Italian, but I studied in France, is biochemistry. And yeah, it just started like that. And I did some, I was going during the summer, I was going to work for free, like to learn in laboratories. And I don't know, I don't know if there is a time when I really discovered my passion for science. I think it was a process, like, just like it started to growing over the years.
No, I totally understand. When you said what you said, it just made so much sense to me. Almost like a process of elimination. You tried different things and then you figured out, oh, this is what, suits me best. But yeah, no, thanks for sharing that. And also, I just wanted to ask about this particular research topic that you will be sharing with us here. And just how did you, arrive at it? What was your process to get to this one? How did your interests develop for this one as well?
Okay, so that's maybe something that I already integrated in my presentation because I thought that, yeah, of course I can present my paper, but my paper is out so everybody can read. So the way I decide to make my presentation, it's about that, like how I did
Oh,
end
okay. Okay.
up doing that, because it's a field that is completely different from what I was doing before. But
Oh, no, no,
yeah,
no.
so
In
I
that
think this.
case, I think we should just go straight into it. Yeah. So you can go ahead and, tell your story the way that you feel comfortable telling it.
And yes, and again, I thought it maybe for the audience would be more interesting to know, like, this is a kind of a chronological order, which is something that we never do. When we write a paper, we always write like in a way that the story is the sexiest as possible. But I thought it would be interesting to see, like, chronologically how I ended up finding what I found. So I guess you guys have the PDF. And yeah, so like I'm going through this PDF right now. And yeah, so my work, the work I've been published, is about this population of CD80 cells that um, are characterized a form of neurodegenerative disease that is called amyotrophic lateral sclerosis 4. And so how I ended up working on that, I am an immunologist and I work in a department of microbiology. And so that my lab is a lab that works like the lab of my mentor, Ivan Marazzi. It's a lab was more focused on discovering epigenetic factors that regulate the inflammation. And so people in the lab, they were screening for these regulators of viral infection using influenza virus. And they ended up discovering that these gene attacks, which encoded for the protein that is called synaptaxin, so the synaptaxin regulates the expression of inflammatory genes during influenza infection. And like I just put one figure showing that when you take cells, you deplete synaptaxin, you do a knockout of synaptaxin, then you infect these cells. You have an increased expression of interferon and IRF3 dependent genes. And that was the story published by the lab. But what is interesting is that these proteins synaptaxin, it has been also linked to neurodegeneration. And so synaptaxin is a protein that is ubiquitously expressed, is an RNA DNA helicase, and it's mainly known because of the role in the resolution of the R loops during transcription. And I mean, there are several, you can see in my slides, like several functions that has been described for this protein, but included what has been shown by the lab of my mentor. It's a regulation of the transcription of inflammatory genes. So it's interestingly, SETX is mutated in two forms of disease, neurologic disease. So there are patients that have mutation that lead to a lack of expression of this protein. And so this patient, they have this disease that is called AOA2 for ataxia ocular apraxia 2. And then there are patients that express a mutated form of this protein. And so in this patient, they develop what is called amyotrophic lateral sclerosis 4, which is a juvenile, slow progressive form of ILS. And so when I joined the lab and then I saw that as an immunologist, I thought it was interesting, this fact that this protein that was just discovered as a regulator of the immune system was also involved in neurodegeneration. So, and I started like trying to understand better this disease, amyotrophic lateral sclerosis. And I have to say, this is not one disease, it's a group of disease, but mainly what they have in common is that you have the generation of motor neurons, which are these neurons that control the voluntary muscle movement in the spinal cord and in the brain. And basically, when you have neurodegeneration of this motor neuron, you have muscle atrophy, and indeed, the patient with ILS, they have like a range of symptoms from difficulty walking, difficulty of speech, swallowing. They experience also a weakness in the upper and or the lower body. And I mean, a lot of work has been done to try to understand also specific mutations that are that cause different forms of ILS. And here I put a table with like all the mutation, the genes that have been discovered so far. And again, as an immunologist, what caught my attention is that several of these genes, they are also involved in the regulation of the immune system. Like, for example, FAST, CDP43, they regulate the NF-κB activation. We have optinurine, sequestosome, TBK1. They are like main regulator of autophagy. C9 has been also been involved to regulate inflammation in microglia and macrophages. And so this to me suggested that there's probably also an immunocomponent in some form of ILS. And indeed, I choose this gene senataxin for the reason that I just explained to you, that is linked to ILS4. And so basically, as I said, this is a juvenile, slow progressive form of ILS. And there are like 3 mutations that have been identified, and the far most common of them is the mutation L389S. But we don't know how this mutation affects the function of the protein and why it causes ILS. But what is interesting is that, what is good for us is that senataxin is very well conserved between human and mice. And so because of that, mice that express exactly the same mutation as human have been generated, that's what I call the SETX knocking mice. And they also have a progressive motor phenotype that you can start to detect when these mice are 8 months of age. And so what I did, I just asked the group that developed these mice, it's a group of Albert Laspada, that now is a UC Irvine. And I got these mice, and my idea was, okay, let's immunophenotype these mice. Let's see if there is any immunodysfunction that can be linked to the disease. And so this is the very first experiment that I did, because I didn't know if, again, there was any role of the immune system in the disease. And I generated bone marrow chimera. So basically what you do, you take the wild type mice, you take these knocking mice, and you lethally irradiate them. So by doing that, you completely deplete their hematopoietic system, and so all the cells of the immune system. And then what you do, you reconstitute these mice with bone marrow cells from either wild type or knock-in mice. And so by doing that, I generated these 4 groups of chimeras. So 2 groups controlled with what I call wild type, that they didn't express the mutation at all, or this knock-in. They were knock-in mice that were reconstituted with bone marrow cells from other knock-in mice. So the mutation is expressed everywhere. But I also had these knock-in wild type mice in which the pathologic form of ZX was expressed only in the immune system and not in the central nervous system, or the wild type knock-in in which the mutation is expressed only in the central nervous system, but not in the hematopoietic system. By the way, guys, feel free to ask me any question if at some point I'm not clear. So I generated these 4 groups of mice, and then I tested them for motor impairment. And first of all, I used the rota rod, with this like kind of a very common way to test motor impairment in mice. So basically, you put the mice on this rod. You have an accelerating rod, and then the length of time that the mice stays on each single mouse stays on the rod, it's like a measure of motor coordination and strength. And so you can see in this graph, I always do 3 trials to be sure that the results are consistent. And you call like latency to fall means you look at how long these mice they stay on the rod. And when we look at the control, that is at the white dot and then the gray dot. So these mice that express these chimeric mice that express the mutation everywhere, they had a motor impairment as compared to the wild type control, and it showed that the radiation and the bone marrow reconstitution did affect the capacity of these mice to develop a phenotype. But the very interesting result was that when the mutation was expressed only in the central nervous system or only in the immune system, the mice they didn't develop a motor impairment. And we confirmed that by looking at the area of motor neuron in the spinal cord and also the number of big axons in the sciatic nerve. And so altogether, these results, they suggested that to develop the disease, at least in mice, you need a factor that you also have you need the contribution of the immune system. And so I try to understand which cell subset of the immune system could be affected by this mutation. And so I started with the innate immune system because, as I told you, everything started when people in the lab, they just discovered that lack of ZX increased the inflammatory response in innate immune cells after infection. So we said, okay, let's see if this mutation L389S recapitulate what we have seen in the knockout cells. And so I used bone marrow-derived dendritic cells. Here infected with Citrobacter rodentium, which is a pathogen, is a bacteria that is a natural rodent pathogen equivalent of E. coli in humans. And what is good is that it induces a high like IRF3 dependent and interferon dependent type 1 interferon response. And so then I compared the knockout cells when they were compared to the control, they express higher level of interferon beta and IL-6, but I didn't see any difference when I compared these knock-in cells to their wild-type control. And I also infect these cells with influenza. I tried to stimulate them with CGAM to stimulate the STIM pathway, and I didn't see any difference in the inflammatory response. And so then I was like, okay, what happened here? I had my chimeric mice, and I was like, okay, let's analyze as immunophenotype the immune system, these chimeric mice, and see what happened. And so I did the CyTOF experiment. So there's this mass cytometry when you can use more than 30 markers at the same time. And I took the blood of this different group of chimeric mice, and I look at different population, innate immunocells, adaptive immunocells, and consistent with what I saw in vitro. I didn't see any particular phenotype in the innate immunocells. But when I look at the T cell compartment, I found that this population of CD8 T cells, so if you, there was a large increase in CD8 T cells that expressed this marker PD1, and this was specifically in the peripheral blood, but I also had data in the central nervous system of the group of chimeric mice that developed the disease. And so I was kind of puzzled at that point because I have to be honest, we really thought that this was an innate immunophenotype, because also everything that showed you at the beginning, all these genes that be associated with ILS, and they mainly control the innate immune system, but not the adaptive immune system. And so I checked in the literature what was known, and I just I found that T cells, actually there are, and especially CD8 T cells, have been shown to really be involved in the brain. They can mediate cognitive dysfunction when upper infection with the neuropathogenic flavivirus. You can have these memory resident T cells in a human brain. They also, CD8 T cells controls the aging and by interacting with the neurogenic niches. And finally, there was this paper about Alzheimer's disease that showed that you have this chronally expanded CD8 T cells population in Alzheimer's patient. And so this made me like more comfortable about my results. So what I did, I went back to my regular mice. These are not chimeric mice. They wild type and knock-in. And I look at the presence of these activated CD8 T cells that express PD1 in different organs and at different age. And I found an increase in these cells statistically significant when I compare knock-in and wild-type mice, specifically in the spinal cord, in the brain, and then in the blood of knock-in mice starting at 6 months of age. In the blood, I can detect this difference a little bit later, 8 months. And you have also FACS plot for people that like FACS, that you see what happened in the spinal cord in 8 months old mice. And I also check other organs. I checked the spleen, I checked the lungs. I didn't see any difference in other organs. So these cells, they were increased only in the central nervous system. And at that time, that for me was kind of interesting, because when I tested this, when I knock-in mice by rotarod, I started to detect a motor impairment when they were 8 months of age. So this suggests to me that this population increased just before these mice that show phenotype. And then it keeps increasing, and this phenotype is stronger in the spinal cord while the disease, as the disease progresses. And so, of course, then the question was like, what are these? I think here I skipped. Sorry, when I did my PowerPoint, I skipped one slide, but it's okay. I was just trying to understand what these PD1 positive cells they were, because PD1 is mainly known as a marker of exhaustion that is not expressed in the functional cells, but indeed these cells, they were functional because in vitro they were able to produce TNF alpha, interferon gamma. And so to better understand the phenotype, I did a sequencing of these cells, and I found that these cells, they were expressing a high level of this transcription factor that is called TOX. And again, why I was puzzled, like, what does it mean? And I went back to the literature, and I found that TOX is highly upregulated in cells that undergo chronic antigenic stimulation. So the T cells, they express this T cell receptor that allow them to recognize a specific antigen. And when the cells they recognize this antigen like over and over again, apparently they upregulate this transcription factor, this TOX. And this has been observed in a human in this paper from Science Immunology when they look at different populations of CD8 cells in the peripheral human blood. And they found that TMRA CD8 cells, which are a population of terminal differentiated CD8 cells, they express high level of TOX and low level of another transcription factor that is TCF1. Now, mice that don't have TMRA, but when I look in the literature about mice, I found that indeed this signature is TOX high, TCF1 low, together with expression of PD1 and another regulatory receptor TIGIT, is characteristic of autoreactive CD8 cells in mice. So in autoreactive CD8 cells, so CD8 cells that react to self-antigen in the central nervous system, but also, for example, in the model of type 1 diabetes. And so I went back to myself, my mice, and I took my symptomatic knocking mice. I look in the spinal cord, in the blood, and I want to see if these PD1 positive CD8 cells, they also express this signature. And indeed, as compared to PD1 negative, they upregulate TOX, TIGIT, and downregulated TCF1. And of course, you have a quantification from different mice. And so far, what my conclusion was that at least in mice, the immunocompartment contributes to motor neuron disease in these SETX L389S animals. And when I was looking for immunodysfunction, what I found was this population of CD8 cells expressing the marker PD1, they were highly expanded specifically in the central nervous system, in the blood of the symptomatic mice. And these cells express marker of cells that are chronically stimulated, like the TMRA in humans and autoreactive T cells. And so at that point, the idea is, okay, are these cells maybe autoreactive? So the cells that react in response to an antigen that is expressed by the host. First of all, to determine that, I needed to know if these cells that can be, they were activated in an antigen-dependent way. And so just like quickly, just to be, what does it mean? So again, when you look at the immune system, you have innate immunocells and adaptive immunocells. And innate immunocells, they're not specific to antigens, but they react to conserved molecules that are expressed by different families of pathogens. T cells and B cells, they have the capability to recognize specific antigens. And in the case of T cells, their main activator is the TCR, for the T cell receptor, which is expressed on this, which actually the TCR is generated when the precursor of T cells are in the thymus. It's generated by random recombination of DNA sequences. And so the TCR, it's in general is unique to each T cell, and it define the antigen specificity. So what happens when you have a function, you have the successful generation of these a functional TCR, then you have these non-activated CD4, CD8 cells that they go to the periphery, and eventually they interact with the innate immunocells that are called APC, antigen-presenting cells. And these cells are responsible for the processing and the presentation of antigenic peptide to the MHC class 2 in the case of CD4, or MHC class 1 in the case of CD8. And so these antigenic peptide, they can derive, of course, from a pathogen. They can derive from proteins that are expressed by the host itself. Or you can have, in the case, for example, of tumors, generation of a completely new antigen. what is interesting here to understand is what is important here to understand is when a T cell express, when a T cell recognize its cognate antigen, it start to actively proliferate. So you have a clonal population of T cells that express the same TCR sequence because they are all generated from the same cells. And why I'm telling all of that? Because the way that we had to understand if these PD-1 positive CD8 T cells were generated in an antigen-dependent way was to look at their TCR sequence. And so I sorted the PD-1 positive and negative CD8 T cells from the central nervous system of symptomatic Noki mice. And then we perform a TCR sequencing, and you have here this bar, each bar is one mouse, and you have like both PD-1 positive and negative, and each color corresponds to one sequence. And you can see that in the PD-1 positive, one or two sequence, they comprise the 60% of the entire repertoire, which is not the case in PD-1 negative. And so this means that when you have the one or two T cell sequences that are overrepresented in a population of T cells, it means that you add like antigen recognition and expansion of the same clone. And we see also the same result in the blood, even if this clonality was not as striking. But more interesting, when we compare the most expressed sequence, TCR sequence in the blood in the center of the system, and we had an overlap between 60 and 80%, meaning that these PD-1 CD8 cells that we also saw in the peripheral blood, they were the same that we could detect in the spinal cord. And so again, the second conclusion is that these cells indeed expand in response to an antigen, but which antigen? And then so this was complicated. So mice are in a pathogen-free facility. So to me, it was not possible that this antigen was derived from a pathogen. And also the fact that these cells, they were found only in the spinal cord, in the brain, and then they were recirculated in the blood, okay, but they were mostly found there, not in other organs. So just to me that if it was an antigen involved, this antigen was probably an antigen that was in the present only in the central nervous system. Now, so these cells, they could have been like autoreactive cells reacting to a CNS-derived antigen. Now, how do you prove that? This is very complicated because it's very difficult to activate ex vivo cells that are specific to a cell-derived antigen. And so I had to be very creative there, and I started looking around, and I thought about the fact that indeed autoimmunity is kind of the other side of the coin of anti-tumor immunity, because when you have a tumor, you want cells that are able to recognize an antigen that is expressed by tumor cells, which are also cells that these cells are derived from the host. And I was looking also at paper in another form of ILS, the C9 ILS, and people suggested that that epidemiological study had shown that these patients, they are more susceptible to autoimmunity and less susceptible to certain kind of cancer. And so what I did then, I tested if my Noki mice that I had this increased population of CD8 cells, they were able to be more resistant to a certain kind of cancer. And so I took this wild type and Noki mice, 1 year old, so the age again that these Noki mice, they were symptomatic. And one group of mice was injected, so I did this injection in the flank because I didn't want any confusing result with what was happening in their central nervous system. So one group was injected with these glioma-derived neurostem cells, And another group was injected with these B16 cancer cells that trigger like that generate melanoma. And at the day that I need to sacrifice the wild-type mice because this glioma was too big, these Noki mice, their tumor was very, very small, and you can see then that I followed the tumor growth over the time. But on the other hand, I didn't see any big difference in the melanoma growth. And I did some histology taking this glioma from the Nokine wild-type mice, and I also did CD80 cell staining. And you can see here in this brown dot, and I found that there was an increased infiltration of CD80 cells in the tumor in Nokine mice as compared to wild-type mice. And again, I also took these cells and we did a TCR sequencing, and it looks like there were some cells that some of the most expressed TCR sequence that were common between the tumor and the PD-1 CD80 cells in the spinal cord. And so that to me suggested that it's a big correlation, but was telling that these PD-1 positive CD80 cells that are expanded in the central nervous system, they are functional and they recognize antigens that are derived from the central nervous system because they can move from the spinal cord and then go and protect the mice against neurostem cells that have tumors. But they don't do the same when you inject them with melanoma. So it means that they are very specific for something that is expressed in the central nervous system. And so that in parallel, when I was having this result, I started wondering if this was true in mice, but what about patient? And so I started collecting peripheral blood from ILS4 patient. And I actually did the same that I did with my chimeric mice. So just here I used cytometry, the Aurora, and I took peripheral blood of this patient and I chose to have healthy control of the same age and the same sex possibly. And again, I immunophenotype, I tried to characterize all the cells, and there was a cluster of this gray, in gray here that really caught my attention. And I look at where these cells that were highly enriched in the ILS4 patient, they were indeed TMRA cells that in humans are CD45-Ra, CD28 negative. So as Noki mice, also ILS4 patient, they were enriched in this population of TMRA CD80 cells. And then we did a single cell sequencing. So we use the site-seq when you can do RNA expression, but you can also use some, you also sequence some antibody on the surface. And actually, we could reconstitute a different population of naive, central memory, effector memory, TMRA CD80 cells. And when you look control and ILS4, there is this population that is highly enriched ILS4 patient that is corresponding to the TMRA CD80 cells. And these cells, they also, we look at their gene expression, they were highly activated. And we also did in parallel the TCR sequencing of these cells. And what we found is that it was this population that we call hyperexpanded cells. So one TCR was expressed by more than 100 cells. And these, when you look at the site-seq profile versus the TCR sequencing profile, these cells that are in red, these hyperexpanded CD80 cells, they were indeed these TMRA CD80 cells that we found in patient. And we also quantified this in you can see in these donuts. We didn't find this hyperexpansion of TMRA CD80 cells in any of the control. Now, this was confirming that we had this kind of the same signature in patient and mice. Now, again, my hypothesis was that these cells, they were autoreactive. But of course, in mice, it's easier because you have these animals that are in the pathogen-free facilities, but in humans, it's kind of complicated. And so what I did, I used this, I bought this like a pool of peptide that are class 1 restricted. What means class 1 restricted is are peptide that can be recognized by specifically by CD8 cells. And these are peptides derived from several. See here the list of the most common pathogens in human. And then I stimulate these cells from peripheral blood, CD8 cells for ILS4 patient and control. And I look at activation of CD8 cells upon stimulation by measuring interferon gamma production. And you can see the production of interferon gamma was similar between control and ILS4 patient, meaning that these highly expanded increased population of CD8 cells that I found in peripheral blood was not specific to any of these pathogens that I tested. And so finally, this was really hard because it's this disease is really rare, but I could manage to find at least 2 post-mortem. There were 2 patients that donate their organs after they died, and so these are ILS4 patients. And we perform a lumbar tissue section from the spinal cord of ILS4 patient. And here we are in the ventral horn. It's the spinal cord where you have the motor neuron that are these big cells. In general, these cells are not in the parenchyma, but in this patient, we found these CD8 cells that were going into the tissue. And we didn't find CD8 cells in the spinal cord of non-ILS patient. And we didn't find these CD8 cells in the parenchyma from, for example, the cortical brain tissue from the same ILS4 patient. And so that was the conclusion of this work. So again, the immune system looks important for the disease in these ILS4 mice, and in these mice we found these TMR-like PD-1 CD8 cells in the center of the system in the blood, and these cells they confer resistance against glioma, suggesting that are probably recognize an antigen that is expressed in the center of the system. I also tested other 2 mouse model of ILS using the SOD1 and the FAST mice, but I didn't see the same phenotype. I didn't see any aberrant CD8 cells expansion in the center of the system, these mice, when they were symptomatic. Now, this could suggest that probably this phenotype is specific to some form of ILS. But I have to say that I am collaborating and talking to some clinicians, and that they also see these CD8 cells in the cerebrospinal fluid of patients that have SOD1 mutation. So I don't know. There can be because sometimes mice they don't match humans, or I don't know. Let's say I'm not pretty confident anymore that this is something specific to ILS4. Let's say like that. But yeah, we got this result in mice. We got an equivalent population of TMR CD8 cells highly expanded in the peripheral blood of patient, and these cells can also be found in the parenchyma of the spinal cord of the same patient. And what I think is the significance of this work, I think, first of all, this is gonna maybe help us to understand the disease mechanism. So diseases are really a complex disease, and until more like 10 years ago, we thought that the central system and the immune system, they were completely separate compartments that were independently, they were completely independent. Now we know that these 2 compartments, they really interact. And so maybe to understand this kind of diseases that are really complicated, we should also try to look at the immune system. As I said at the beginning, it's a group of heterogeneous disease. And so I think it's very important to understand what is common to this disease to find a therapy for everybody. But it's also important to understand what is different, what is peculiar to different form of ALS for the purpose of diagnosis, but also because like this we can tailor our treatment in the way that are going to be more efficient in different groups of patients. And finally, this is the model that I'm working on, and so this is my I drew by this one myself. So these are my motor neurons on the left that are healthy, and then if they express this attacks L389S, you see now that they become pretty ugly, are these like kind of brownish motor neurons. And I think this is the first step of the disease. I think the disease started with the neurodegeneration of motor neurons, because if you take this motor neuron from leukemia mice, you take the neurons from leukemia mice and wild-type mice that you put in culture, the leukemia neurons, they are more prone to die. So there is something intrinsic in these motor neurons that this mutation make them more fragile. But then at the same time, something happened, maybe there is an inflammatory environment, maybe there are other cells like microglia involved, you have this proliferation, this activation of CD8 T cells that are these round cells in green. And what is interesting, and while these cells expand, and then you have a progression of the disease while these CD8 T cells expand. Now, how these CD8 T cells are doing and what is their role specifically in the disease, it's something that I'm really trying to investigate right now. And then I put just a last slide of acknowledgement, because of course I had the help from a lot of people from my lab, Albert Laspada's group. They helped me with, they give me the mice, and I have to collaborate with people, with clinicians for the tissues, but also like to a patient, and yeah, there is we have collaborator, Sean Chisari is the second author of my paper, and he did the single cell analysis. This is a guy, he was first year PhD student, so amazing guy, and okay, the funding. Thank you, and I'm open to any kind of question.
Thank you so much, Laura, for that wonderful presentation. I'm pretty sure a lot of people on stage and in the audience have a lot of questions for you. But I saw you unmuted, Katerina. Do you have something?
So such interesting work. I really always had I had this theory for like years that the communication between the neural system and the immune system is very important. That turns disease into a chronic state, and then it's very so this work is fascinating, and yeah, it makes me really happy that you got funding for this and you went for this. So congratulations, Laura.
Yeah, and I have to say, just it was pretty hard, because that's true that now this is start to be more a trend, like the T cells and neurodegenerative disease. But when I started this project, people, they thought I was completely crazy, and it was really difficult to get this project, to get funds for this project. We got this CZI from the Zuckerberg Foundation, that it was actually kind of easy to get, because it was a foundation and they like just to have really bold, crazy project, because yeah, those are just like to tell you guys, don't get discouraged, because just go and with your data, because it's true that sometimes people don't believe you, or don't think that what you do is interesting, but sometimes they are just wrong.
I like that. Sometimes they're just wrong. Thank you. All right. Jamie, do you have any questions?
I had this one for you. It's very interesting when you were telling about your discoveries here. Did you have any opportunity to examine this in the mice through like hereditary, like any like babies or anything like that? Did they have the same results to it? Was anything passed on, or maybe you didn't have a chance to do that in this particular study?
So can you repeat? I think I didn't understand.
Oh,
Which mice?
I beg your pardon.
Oh.
I was wondering with all of the findings that you were describing to us, if you had the chance with the mice to see how any of this is passed on through inheritance with the babies or anything, if you saw the exact same sort of symptoms in the baby mice or anything like that, or perhaps that might be a different study. So.
No, I mean this is familiar form of ILS. So in both mice and humans. So this is a so it goes through like it is our autosomal dominant. So the progeny has unfortunately who get this mutation uh, get sick. This phenotype is not found in very young mice because uh, the mice that start to get sick when they are like 8 months of age. And I detect these cells when the mice are like 6 months of age. But I analyzed younger mice and I didn't see that. So I think this is not something that this is something that is just like, as I can say, is a progressive phenotype. And so probably a very young mice, there is no difference between the ILS for mice and the wild type mice. If I did I get your question?
Yeah, that was it. Thank you very much for indulging me.
Thank you.
Thank you.
All right. Denise, do you have any questions?
Yes, thank you so much for this amazing presentation. I was struck immediately by this being influenza, and given that there is another pathogen in the family Coronaviridae currently wreaking havoc on this planet, I was curious if there were any implications for the SARS-2 pandemic, your findings in that disease.
I mean, it would be interesting to understand if these patients are more susceptible or not to virus, but to viral infection, whatever infection. I mean, the patient that I have so far, the group is too. I have to. There are so few patients I cannot conclude. What I can tell you is that when you don't express these proteins and a taxin, you have a higher expression of inflammatory genes. So based on like I did some studies in COVID, like mice SARS-CoV-2, and the problem is that in there is that the inflammatory response at some point become too you have an exaggerated inflammatory response that what makes you sick, what like kind of damage also the organs of the host. So if this protein plays a role, it would not be good, like because this protein, if it's not expressed, then inflammation is too acute. It's too you have too much inflammatory. You have the expression of inflammatory genes is too high, so it could be damaging. So if I would guess, but in regular people, SATX is functional, so we are able to regulate our inflammatory response.
Got it. Thank you so much for your answer.
Thank you.
Dr. Shah.
Yeah, thank you so much, Laura. That was a fascinating work. I was a little bit late. My question from you, because you mentioned about the interferon gamma, and it seems that you didn't find the, significant relationship between your pathogens and the interferon gamma. However, we know that the level of the interferon gamma, beside the, for example, some of the factor as a pro-inflammatory IL-6, IL-8, or TNF. So it's gonna elevate it in patient, and some of the patients, based upon the type of the ALS, which is that spinal or bulbar, from the starting point, has a different result. I was just wondering because it's an animal study, and I wanna hear what is your thought around that.
So I mean, I don't know which experiment were you referring at, because I mean in mice, so these cells, they don't secrete just interferon gamma, they express interferon gamma, TNF alpha, granzyme. So the experiment that I did in vitro with the peripheral blood of ILS4 patient, the readout was interferon gamma, but it was just a readout for CD8 cells activation, just to see if the response against pathogen was higher in patient than in the control, which was not the case. Then, if you are asking me about like what exactly the function, the effector function of these cells, I agree with you. I think that it's probably not restricted to interferon gamma, but this is what just a readout. You can look at IL-2 just to see if the cells are getting activated ex vivo, but in vivo is different. Yeah, I agree.
And we are talking about the peripheral adaptive immunity, right? Specifically when we are talking about IFN gamma. I want to be sure that you have emphasized on the peripheral adaptive immunity.
Yeah.
And also, did you just had elevated on a CPR and all of those inflammation factors or markers?
If I have, sorry?
For other inflammatory factors like a CPR and all of those elements, ESR and all of those things, did you have any result from that part as well?
No, I just look at the T cell phenotype, and then I look at that. We did a single cell sequencing of these CD8 cells, so they express like granzyme, perforin, marker of activation, but just like we look just on CD8 cells.
Thank you so much, Laura.
Thank you.
All right, Gilbert. Gilbert, are you here? Okay, then I guess Victoria. Hi.
Hi, I just wanted to say thank you for a very lovely presentation. And I'm curious because you are connected to the microbiology department, if you explored differences in the microbiome of the mice that you studied to see if there were any discernible differences there.
I didn't, but I agree with you that would be interesting. Yeah, I didn't, but that's true. That's at some point we thought about that. Yeah. Sorry, I don't have an answer, but yeah, this is something you're completely right. It would be interesting to look at that.
All right then. Does anyone else in the audience or on stage have any questions for Laura? Please post your questions in the chat if you don't want to come up on stage, or you can raise your hand and come up on stage and ask your question.
I guess I have one follow-on question, if it's okay.
Oh, no,
I'm
go
just
ahead.
curious, Laura, what do you think the implications are for the basically for future treatments based on the findings of your study?
I mean, and again, like there are a lot of like work that needs to be done, but let's say if there is a role of the cells, I think that could be also good news in the sense that you can manipulate the cells. I think it's easier to manipulate the cells than neurons. For example, you can deplete a population of CD8 cells, and of course you cannot deplete a population of neurons. So there is there are a lot of so molecules that you can use. So, but also I think like more in general, it would be like important to think about like different factors that can participate to the disease. I'm saying that because I met with clinicians that are treating SOD1 or FAST patient with oligoantisense, that you inject this oligo, you try to correct the genetic defect. And they told me that the results are kind of partial, because they believe that what happened is that you start with the genetic defect and these, for example, motor neurons, but then after that you activate a cascade of events. And so maybe they were saying, maybe if we are able to combine different kind of treatment that are maybe genetic therapies together with an anti-inflammatory, or by targeting some particular population of the immune system, that's something that we could get a better result. So I think that there are two ways to see that, to look at that. The way, like, really trying to see if the T cells plays a role, because these cells can be targeted easily, but also try to understand when we do a therapy for this patient, that's probably we need to target more than one cell subset to get result. Thank you.
All right. Does anyone else have any other questions?
Yeah, thank you so much for answering all of that. I wanted to ask, so how good will you be able to use this also as an early diagnosis? Is that also something you can do maybe pretty early, like pretty soon?
Yeah, this is it's a good question. I now I don't know, because I had I didn't have patient young enough, but of course this in the one idea is because also this is a form of ALS that is progressive, so you have also patients that are kids or teenagers. So the idea, it would be to look at that. So if there is, in mice, we know that there is a correlation, like between these cells that increase before the symptoms, and then you have a correlation between the number of these cells and how much it gets, the symptoms of these mice, how strong they are, the symptoms. In humans, so far, I don't know, but it would be interesting if I can have enough patient at different stage, from different age, to look at that. Yeah.
Yeah, that's interesting because we also had the guest speaker. She found a T cell activity difference in Alzheimer's. So maybe it could be, a more broad, applied principle basically to, diagnose early and then also deliver some sort of treatment um, that way. So.
No, that you're right, because in this paper on Alzheimer's disease, they see that again, the increased number of there was a positive correlation between the number of these TMRA CD8 T cells and the loss of cognitive function. So again, these were patients that they use in this Alzheimer's this paper, that when you have a sporadic disease is kind of tricky to understand. You have the patient when they are already pretty sick. Now, are these cells present in pre-symptomatic Alzheimer's patient? Can we detect any, for example, susceptibility? That would be a good question, but yes, I think it would be difficult to assess, at least in humans, but we can start with mice and see.
Yeah, the other question is if you think, sorry, I'm with my kids in the car. The other thing is if there's a predisposition, do you think there would be one?
Sorry, I think I cannot hear you anymore. There's something.
Yeah, you muted there, Katerina, for a second. You cut out. Yeah.
That may indicate she's having connectivity issues.
I thought she had asked if
Can
there was
you?
any predisposition.
Yeah, exactly. Yeah, so if you can detect in these patients in the T cells maybe a vulnerability of overreacting basically and contributing to this neurodegeneration.
Yeah, no, I see what you mean. Yeah, that's something that I think is maybe more feasible in when you have a familiar form of this disease, like test what you're saying. Probably we should start with people that have a familiar form and see like in a family with their history of ALS or Alzheimer's disease, if there is any specific marker in their CD8 cells, and that this is gonna predict to have a predisposition to disease. Yes, definitely. That would be great because then you can also predict in people that don't have a familiar history of neurodegenerative disease. Yeah.
Yeah, the other thing would, if the other thing would make this interesting in case there's a predisposition or not is that maybe you could find I don't know, but maybe for example, if people that have a high vulnerability for these disorders, probably an occurrence of like a severe inflammation through viral infection or any kind of bacterial infection would be really bad for them. So it would be maybe, if you could do this pre-screening pretty early on in life, you could maybe suppress the immune system during those stressors that would make them have maybe the disease progress faster or an early onset or maybe delay it more. That's my thought for the
No, no,
future.
no. I think you are right, and I think in the at least in the field of the Alzheimer's disease, now these labs they are trying to understand if there is a link between infection and the CD8 T cell response and Alzheimer's disease and this kind of thing. But yeah, and I'm trying to understand if there is anything like specific, like there's a what is wrong with CD8 T cells, and then I agree and see if there is any other component like infection or like we were saying the microbiome that can affect the cells in the disease, and so can we prevent some of that? Because, yeah, can we do something about to prevent the disease? Yeah.
Yeah, thank you so much. We are around an hour, like we went around an hour. So yeah, I wanted to ask, how much time we have left if you need to basically leave the room. And yeah, thank you so much.
I mean, if there is any further question, I'm happy to answer. Like,
Yeah, I can go on with questions if nobody else has one. Maybe I'll ask another one and then. So what are you planning right now next, or probably you're already working on the next step? What's in the future?
yeah, I think I'm really, really curious about the role of these cells. If they really like have a role, they are a marker of disease, if they really participate actively to the disease. There's one thing, and I'm interested in that these cells they can be also found in other form of ILS.
So if did you try to do culture maybe with motor neurons that, you know, have basically start the damage, and then you know, have them in a basically in the T cell environment that is kind of regular. I don't know if that works at all, to see basically if healthy, normal, I don't know if that even is a thing, T cells can basically stop the progress, like if the neurodegeneration slows down or stops in a healthy environment, basically.
Yeah, this is interesting. Yeah, it's something that I would like to do. I need to deal like with the very few these cells there. In mice, at least, like the number of these cells is not great, and they look pretty fragile. Like when you take out from the mouse. I mean, I think this is something that you have with the T cells that are in the tissue. They are not very good surviving after, but yeah, definitely this is something that I really want to, I already want to do before sending the paper. And then I tried a couple of things that didn't work really well, but this is something that is worth like troubleshooting and doing. Yeah, I didn't try, but this is a fantastic idea. Yeah.
Yeah, maybe in a humanized immune system mouse model that you can out of the disease model regular. I'm not sure if it will work, but this humanized immune model, and then see if it works. Or I don't know if you did you get hands on human
You're in the matrix, Katerina.
like, is it
Sorry, I couldn't hear well.
No,
basically designed the experiment, if you can hear me, but
a little bit better.
my best. Yeah, can
No, you've gone out again. It's not just me, right? She's
No,
No,
I
deep
okay.
cannot
deep
hear.
matrix.
Okay. If you can, Katerina, maybe type your question in the chat, and we can just read it out.
Yeah, her question is
Okay, I see. Use mouse model with human immune system. Yes and no, in the sense that these mice, their central nervous system is still a murine central nervous system. So the problem is that it's difficult to model interaction between the central nervous system, its mouse, and if the immune system, it's human, because you also have a mismatch, like antigen presentation and this kind of thing is not gonna really work if for immunized mice you mean this. I mean, the immunized mice basically are this is the mice that are highly deficient for cells of the immune system, in cells of the immune system, and then you reconstitute them. So yeah, but but you can still like play, like you can do IPS in vitro, for example, or for and do this. Yeah, so yeah, I think, yeah, if you irradiate, that's the thing, like if you can also irradiate the immune system, the point is that the center of the system is still the murine one, so that you're gonna have a kind of a mouse in which you have a human immune system and the mouse center of the system, and they not gonna talk to each other because they're not gonna be able to do that because there is a mismatch. But I think, yeah, you can do in vitro, for example, do motor neurons derived from, I don't know, IPS. I don't know if this is possible. For sure, you can do neuron from IPS and then incubate this with the cells from a patient. That's definitely is feasible. Yeah, or do like motor neuron from mice, incubate them with the cells from mice. Yeah.
Yeah, thank you. I don't know if it's gone. I hope
Sorry, Katerina, it hasn't gotten any better. Maybe the chat is the best option.
Yeah, no, we can't hear you at all.
Okay. Thank you, Katarina. Thank you very much.
All right. I mean, all right. I guess she's just signing off for now. All right. So I don't know. Does anybody else have any last questions? Because I think Laura's time might be up. Yeah. Oh,
I
Jamie?
have no more questions, but I'd like to just thank you very, very much for your time and a very fascinating talk. And I hope you can actually come back again sometime with any new findings that you have for us. We'd really appreciate that. It's been a pleasure having you, Doctor.
Great, thank you. Me too. I hope I'm gonna have more funding, but again, I really appreciate, guys, your interest and passion for science, and thank you for having me here. It was really a pleasure.
Yeah, thanks for coming. Thank you for taking the time. We always like to have you back. Katerina, have you worked out the issue?
Yeah, thank you so much. If you can hear me, I wish
Yeah,
you,
yeah, much better
all the
now.
best. Perfect, all the best for your future. And I'm very curious to, follow along your research. It's really so. Thank you so much, Laura.
Yeah, all the best for your future, guys, for sure. Like, you are very, very great. It was very great to talk to you. Like, fantastic question and comments. So thank you and good night.
Very good. Oh, and we
Thank
also
you. Have a
we
great rest
also
of your
have to
day.
wish you all the funding as well, because that's what we do at Science Science. We do
Yeah,
everything
for
to
sure.
fund all the funding. So thank you
Absolutely.
very much.
Thank you.
Thank you. Have a great evening, everybody. Good night.
Thank you.
Bye.
Bye.
Bye
Thank you so
everyone.
much.
Bye
Thanks.
bye.
Yeah.
Bye, Laura.
Ending the room in 3, 2, 1.